Okay, thanks very much for that, Mr Griffin. We're going to run through the things that we've covered in the last semester and a little bit of extra as work because we'll be covering global systems as well. I'll be presenting first with basic introductory information about exam venues and preparation and so on. Ms Barker will take over and do the current topic of origins of the universe. I'll get back on and do genetics and evolution, and Mr Ho will upstage us all with his PowerPoint on investigations and on global systems. We'll go until the bell, and then you'll receive a presentation on the CUPS campaign from the girls who have been involved in doing that. This is going to go on the wiki as soon as I get a moment this afternoon, which should be at around 2:30, 3 o'clock. So this will be available for you to download and use at your leisure if it forms part of your revision notes or if it's a reminder. Whatever it is that's useful to you is what it is to you. Yep, right. Ignore the day, it's the date, sorry, Thursday the 13th. If you're in Miss Jack's or Mr Ho's classes, you'll be in DLC level 1. It helps if you look at what set number you're in as well. If you're in Mr Chan's, Mrs Murray's or Mr Heath's classes, then you'll be in DLC level 2. Mr Tez and Ms Barker, Centenary 3.2 and 3.3. The rooms will be open so that there will be a wall open between the two. Myself and Mr Lyons' classes, Centenary 3.4 and 3.5, and Ms Warner's 2 classes, Centenary 3.10 and 3.11. Given that there are 3 venues within Centenary Building and VCAR and IB exams on at around this time, please do pay attention to what signs say no entry, and if you need to go up the fire escape or the external access, then you need to. You probably won't be able to go through the front door of the Centenary Building due to the public exams that are on. What should you bring? Same as you have for the Semester 1 exam. Pens, pencil, please make sure your pens work. Ruler, star card, please, and if you somehow manage to lose it, please get a printout of your information from my MLC. You are allowed to bring a calculator, but only if it is a scientific calculator. You were book listed these for this year, and you had the same regulation due for the Semester 1 exams as well. So same calculator, not the big graphics ones, you won't be allowed to take it in. The Semester 2 topics that will be assessed: investigations, global systems, genetics and evolution, as well as the current origins of the universe topic. There are 2 booklets. Booklet 1 is multiple choice questions. Booklet 2 is the answer page for the multiple choice as well as the short answer questions. You can do this in any order that you like. You don't have to start with booklet 1 and progress to booklet 2. You can jump back and forth as you so desire. The question breakdown: 30 multiple choice questions, and half of them are on genetics and evolution. You don't need to take photos of this because it is going up on the wiki and you can have the real thing. The rest of the breakdown is there, and booklet 2 simply effectively doubles those marks. So half of booklet 2 is on genetics and evolution, and the rest is as allocated. That should take you about 60 minutes, but like I said, you can take whatever order you like, so you'll probably lose track of that. Roughly 1 mark per minute, 90 marks, 90 minutes. That's a good guide. If you start spending 10 minutes on one multiple choice question, you will have a problem at the other end of the exam. The cover page for Semester 2's Booklet 2 will look like this, or similar to this. Rather than asking you to write A, B, C or D, we're offering you the opportunity to colour in, circle A or B or C or D. This is how your senior school exams will appear, such as the VCAR or the IB paper exams. So we might as well start showing you how to do that now. It's the same thing as you would have done for the maths and science competition exams anyway. There's one thing that I'm going to mention before I pass over to Ms Barker, and that is hashtag marks. Because before hashtag, that used to be number. So number of marks is an important consideration when you're responding to a question. If you see 4 marks, how many things do you think you should write about? 4. If 5, if 6, then great. Just not 3 or 2 or 1. You'll not get 4 marks for writing one sentence, I guarantee you that. If you look through the question and short answers and you see the word suggest or describe or distinguish, you are being asked a question which is of medium difficulty, which will have 1, maybe 2 marks given to it. If you see the word list or state or name, you are doing a very simple thing. You don't need to write much, you won't get much credit for that. It will be a 1 mark answer. If you see the word explain or predict, you need to make sure you include how or why does something work. And I will give you a worked example of that for natural selection later on when I talk to you again. Everything else, the lunchtime help sessions, we're going to these as teachers. We'd love to see as many of you at all of them as possible. Please don't leave it until Monday the 10th. If you want time to speak to us, earlier is better. If you feel that you come to Wednesday and Thursday and that's enough, then brilliant. If you want to come to all of them, then you are most welcome. Again, don't take photos of this because it will be on the wiki. Thanks. Preparation is as for semester 1, and I'm actually going to skim over this in the interest of time. So this will all be information that you can read on your own that I can't really contribute any more detail to. To look, objectives, linking to what sort of learner you are with relation to visual or kinesthetic or so on. Active revision, same as I said last time, make your brain work. Don't just read stuff, make sure you present some other form of information. Don't just read over your notes, that it will not help. And do a practice exam. Last year's is on there. Last year's was the same topics as this year's. So make sure that you familiarise yourself with the style of questions by doing last year's exam. Origins of the Universe is up now, so I'd like you to welcome Miss Barker, and she's going to take you through that.
Okay, I'm going to try and get through this quite quickly because I understand that it is fresh in your minds. You've just completed or will be completing the test for this in period 4 today, so we probably don't need to spend as much time on Origins of the Universe as you may want to spend on some of the earlier topics. Okay, so our key ideas in the Origins of the Universe topic are these things listed. So what can we see in our night sky, what is in our solar system, and then a little bit further afield as well, what else is out there. So some stars, bigger things, some galaxies, and how those things are clustered together, how they look in the night sky. It's actually kind of interesting that from a really long way away, a galaxy looks just the same as a star that's close by. Okay, that it's just light. All we can collect from our universe is light, and that's how we do our science for astrophysics. We do often like to know how far away things are, and we have a few different ways of measuring that. We will be looking at the Big Bang Theory and some other theories of how the universe evolved. The Big Bang Theory obviously is the most important one. It's the currently accepted model for the development of the universe. We need to look at what light can tell us. As it's our only source of data for this branch of science, we do need to really look at it very, very closely and ascertain everything we can from that light. You also need to know about the life cycle of stars and a little bit about what might happen to the Earth. Not immediately. For the present moment we're fine, but in a couple of billion years, maybe not. Okay, so in our night sky we can see a whole bunch of stuff. We can see stars, planets, constellations, galaxies, and if you've got just your eyes, you can see Earth's moon. If you've got a telescope, you can see some other moons around other planets as well. In terms of what you need to know about our solar system, you need to know what the planets are. You need to know them in order, and it's probably a good idea to know a little bit about each of the planets. It doesn't need to be a lot, just something about each of the planets so that you can distinguish it from the others. You do need to know where the asteroid belt is in our solar system. It's often something that gets overlooked. There is a big belt of asteroids in between the orbits of Mars and Jupiter. And that funny list of letters that I've got on the board on the screen, is the planets. Now, you would be aware from your lessons over the last 3 or 4 weeks that there has been some debate recently about the status of the furthest planet from our sun, Pluto. It was declassified as a planet a few years ago, and there's been some recent push from certain astrophysicists around the world to have it reinstated as a planet. I don't actually mind whether you call it a planet or not. Different people will say different things. Older textbooks will have it listed as a planet, newer textbooks won't. We know it's an object out there, that's all that matters. Okay, those planets, this is 2 ways that we can try to remember them. This is the one that I got taught when I was a student, your age at school. So if we take just the first letter of all of the planets, we can make a little saying. My Very Easy Memory Jingle Seems Useful Naming Planets. Okay, and that lists all the planets in order. Or, when Pluto got declassified, we simply changed it to My Very Easy Memory Jingle Seems Useless Now. Okay, so that's something that you can, it's probably quite easy to remember. It's what we would call a memory mnemonic. Often it's easier to remember things if we've got some sort of jingle to go with it, or something to identify each of the individual parts. Okay, moving on. Measuring distances. Okay, measuring distances is something that's quite difficult in astrophysics. We can't physically go out there, get a tape measure, stretch it from here to the moon to find out how far away it is. There is simply not a long enough tape measure in existence to do that. The only way that we can find out is to look at the light. Okay, so one of the things that we can look for is stellar parallax. Okay, so due to the Earth's motion around the Sun, for some stars, not all stars, for some very nearby stars, we can notice throughout the year that they seem to move a little bit relative to the background stars. So, for example, in January, we would see that yellow star, which I've called star X, we would see it in between star B and star C. But then 6 months later, in June, the Earth has moved around its orbit, and we would see star X in between stars A and B. Okay, so from our position, from what we can see, it seems as though star X has moved back and forth across star B. Okay, so that is a process called stellar parallax. It actually, people predicted this a long time before we were able to actually use it to calculate the distance to stars. We couldn't actually use stellar parallax until we developed telescopes accurate enough to measure very, very small changes in the direction that we were looking to see those stars. Because when I say they move, I've very much exaggerated it on the screen. They move a tiny, tiny, tiny amount. If you imagine, if you look around in a circle, makes 360 degrees if you look all the way around. The amount that a star would move when we're looking at its stellar parallax is not even 1 degree. It's not 1/60th of a degree. It's 1/3600th of a degree that it would move. Okay, so it's a very, very tiny movement. Okay, we can detect that. Okay, moving on. Big Bang Theory. There's a few main ones that have been popular throughout time, or throughout human understanding. The geocentric one was the first one we came up with. We're very self-centred as a human race. We figured that the place where we live is special, we must be the centre of the universe, and so the geocentric model was the first one. The second one, we got a little bit off our pedestal, and we said, well, maybe we're not the centre, maybe our sun is at the centre of the universe, and so we developed the heliocentric model. Following that, there were two competing theories, the steady state theory and the Big Bang theory. People didn't like the Big Bang theory at first. They didn't believe that the universe could have possibly started as a very small hot point, essentially. And it wasn't until, again, we developed the technology to detect a whole bunch of stuff predicted by the Big Bang theory that we started to take it more seriously. The steady state theory, for those that are interested, just says that the universe is infinite and it stays approximately the same forever. It does expand though, so the steady state theory does include expansion, and then as the universe expands, new matter would sort of pop into existence to fill up the spaces that were created due to the expansion. The steady state theory is pretty much discredited now, so we don't give it a whole lot of time, but the Big Bang theory we definitely do need to know about. For the Big Bang theory, hopefully you would be aware now that the universe did start as a very, very small hot point. So all of the mass and all of the matter and space in the universe was originally compressed into an infinitely small point, and for some reason that we don't try to explain, for some reason that very dense hot point started to expand, and it has continued expanding until it has reached its current state, and it is still expanding. Okay, so how do we know that this has happened? There's two things that we look for to provide evidence for the Big Bang theory. First, we look for redshift. Now, when we look at the stars, we can collect a whole lot of light from them, and some of that light is visible light. And so that means when you see a rainbow in the sky, that's the visible spectrum, that's visible light. The reason that you can see everything in the room around you is due to the visible light part of the spectrum, but there are other parts. For what we're doing, I'm just going to put the visible part up because we don't have the ability to see your ultraviolet light or infrared light or microwave radiation. We can't see that with our eyes. We don't have the right sorts of sensors in our eyes to detect that. So, for what we're looking at, we'll stick to the visible light. And if you can see up on the screen, there's a whole bunch of little dark lines in the spectrum that's at the top. And what we mean by redshift is that the positions where we see those dark lines seems to have shifted towards the red end of the spectrum. Okay, that's what redshift is, and that is a direct result of what we call the Doppler effect and the expansion of the universe. The Doppler effect is when waves bunch up or spread out depending on whether the source of the wave is moving towards you or away from you. In the case of the stars and the galaxies that we see in our sky, they're mostly all moving away from us. So for all of the stars that we look at, we see their spectrum is red-shifted. What we also know is that the further away they are, the more they are red-shifted. Okay, so they'll be moved even further towards the red end for stars that are very far away, due to the fact that they're moving away from us very fast. We also look for the cosmic microwave background. That's some remnants of very hot radiation from the very few moments after the Big Bang occurred. Okay, so we've already covered the stellar spectra, so I'll skip over that one. So the stellar spectra can also tell us what the star is made of, how far away it is, how much light it emits, and how hot the star is. Okay, stars that are red are cooler stars, stars that are blue are very hot stars. All of the stars can be plotted on a Hertzsprung-Russell diagram. That's a diagram that plots essentially brightness on the vertical axis. Now that might say luminosity, it might say absolute magnitude, but it's really about the brightness. Bright stars at the top, dim stars at the bottom. And then on the horizontal axis, we've got temperature or colour. So our blue stars, which are our hottest stars, are towards the left side of the diagram, and the coolest stars, the red stars, are on the right. Okay, so I did mention brightness and magnitude. We have two magnitude scales. Apparent magnitude just tells us how bright the star looks from Earth. Okay, so when we look into the sky, we can see some stars that are very bright. They have a very low magnitude, or potentially even a negative magnitude. So very bright, negative apparent magnitude, very dim, big positive numbers. For the absolute magnitude scale, it's a bit more fair. Okay, we imagine that all the stars could be viewed at the same distance away from us. That distance happens to be 32.6 light years. And so if we could put all of the stars at exactly that distance, the absolute magnitude scale tells us how bright they would look relative to each other. So again, the very brightest stars would have negative magnitudes. Increasing from there, we get dimmer and dimmer stars. Okay, stellar life cycle, we're almost done. When a star forms, a gas cloud collapses, the cloud gets hot, eventually fusion starts in the core. Depending on how much mass there was in that cloud, so how much gas was in the cloud, the star will either be hot and blue, or it will be cooler and more of a red or a yellow colour. As long as the star continues to burn hydrogen in its core, it is what we call a main sequence star. Okay, that lifetime can last for 10 billion years if it's a star like our sun, or if it's a really big hot star, that period of time will be much shorter. Big stars burn their fuel really quickly, and so they tend to change much more quickly. Following the main sequence phase, your stars will move into either a red giant or a red supergiant phase. Following that, there's usually a spectacular sort of explosion or just a gradual loss of mass of gas, essentially. So that's a really hot gas sort of floating away from the star, or a big explosion if it's a heavier star. Those two things are called planetary nebula or supernova. And then following that, we get a few other stages. So a supernova can either form a neutron star or a black hole, and then a planetary nebula will leave a white dwarf in the centre, which is actually technically not a star because there's no fusion happening in its core. It is very, very bright because it's very, very hot, but it's also quite small. Okay, so it might not look very bright in the sky. Okay, and it will gradually cool down, and eventually it will fade from our vision. Okay, and finally, Earth's fate. When our sun runs out of fuel, it will expand to become a red giant, and that star, our star at that point, will engulf the Earth. and life on Earth will be extinguished. There is no getting away from that fact. But don't worry, it's not happening anytime soon. Okay, 5 billion years is about how long the sun has burnt for so far. It's got another 5 billion years to go. Okay, so we're safe. All right, I'll now pass back to Mr Seletto to talk to you about genetics.
Okay. Thanks, girls. Now it's my turn to talk to you about genetics as well as evolution. We grouped those two topics a couple of years ago because understanding how we see variation that we can study at a genetic level also allows us to study the potential for natural selection at a species level. So the two topics do actually complement each other, which is why we did the two last term. It was a very busy term last term because we also had your investigations at the start of it, and there were some classes lost, so it was very, very busy. I'm not going to attempt to reteach you everything this afternoon though. Now, all of this information you see on the slides, as I said, will be available to you. So some things are simply best read later, and there's not much that I can add to it. Where there are things that I need to say, I'll stop and talk through. Biology is effectively the closest to a language that science will get, in that you're not doing chemical formulas, you're not manipulating mathematical formulas, you're using worded answers with words in them that have very specific meanings, such as the word respiration in senior biology, which some of you might think of as breathing in and out, whereas it is actually a cellular process that occurs in organelles called mitochondria. Now, if you misunderstand one word, your response using that word may not be correct. For example, if you use the word gene instead of the words allele, because they are not the same thing, your response will be taken at face value and may not in fact be correct. So be very careful. There are vocabulary resources available in Lesson 1's resources on the wiki. Mitosis and meiosis I will simply cover in a couple of brief points. Mitosis is how normal body cells reproduce to make more cells of you. The two cells that are produced are genetically identical to each other and to the cell that was at the start. Meiosis is the process that converts what we call a germline cell, which is a cell in your body that is going to become a gamete one day, converts that cell to four gametes. Gametes are otherwise known as sex cells. For females, they are egg cells. For males, they are sperm cells. You end up, instead of two cells with four, instead of identical cells, you have four cells with half as much DNA as an original human body cell would have. If we normally have 46 chromosomes, how many does a gamete have? 23, because two halves make a whole. Sperm cell from dad, egg cell from mum, 23 plus 23 restores 46. That's a normal diploid cell. That cell can go on and do mitosis, and here we all are. DNA is a large molecule. It is made up of lots of smaller building blocks. Each building block is called a nucleotide. You should know how to draw these shapes. Do not be any fancier than that. Simply a circle, diagonal line, pentagon, horizontal line, rectangle, and label them appropriately, and you have just drawn a nucleotide. I can draw things in biology, I can't draw things in art. You don't need to be very good at drawing, you just need to be very clear at drawing. On the right of that shows how these things are connected up. We know that adenine, the base adenine pairs with the base thymine, and guanine pairs with cytosine. So the green box on that diagram on the left represents the variable base. It's either adenine or guanine or cytosine or thymine. If you are looking at a molecule of RNA, you will not find any thymine. Instead, you will find uracil, which means adenine pairs with uracil in RNA, but in DNA, A with T, G with C. If we start at a larger level and look down, you look through a microscope at a cell, you see the nucleus. You look closer at the nucleus, you see strands of DNA. Those strands are called chromosomes, and a human cell has 46 of them. You start to look closer at a genetic level, and you can see that there are sections that don't look any different in terms of colour or shape or structure, but are the important sections of information that matter when your cells want to do something, like make an enzyme or carry out a growth process or whatever. The sections of a chromosome that actually matter, the instructions to make us, are called genes. We have around 25,000 of them, and genes are the instructions for making molecules called proteins. If the gene sequence changes through some form of radiation like UV or overexposure to X-rays, or it could be some other mutation, then the protein that's produced as a result will be slightly different and may not function and do its job anymore. That would be what we call a gene mutation. The other type of mutation that you've done relates to an example most commonly used for this, which is Down syndrome. A person with Down syndrome has inherited 3 copies of chromosome 21. So one of them has come from one parent, the other parent has given them 2, which means one normal one and an extra one as well. That's a whole chromosome mutation. The gene sequence is perfect, there's just an extra chromosome that shouldn't be there in the first place. The reason it's there is because of a mistake in meiosis. You can look at a diagram called a karyotype. A karyotype is when you take a cell, take the DNA from the cell, arrange it visually, and that is what you get, which allows you to tell how many chromosomes are present. For humans, we expect to see 46. If there is an extra one, we can figure out which one it is. It also allows you to see if the person has 2 X chromosomes or if they have an X and a Y, which means XY is male, XX is female. Variation I'm going to leave. The only thing I want to say is that if we don't have any variation in a population, natural selection can't work. If we are all the same, either we all survive or we all don't survive. Alleles is the most commonly misused term in genetics. I talked about a gene being an instruction for a protein. The allele is the version of that gene. For example, there is one gene that controls your blood type. There are 3 alleles for this gene. One is A, one is B, and one is O. One gene, multiple alleles. If you look at coat colour in mice, or you look at haemophilia, there's one gene, but there are different versions of that sequence which result in different phenotypes, which is what we describe you as having. You have haemophilia, or you are colourblind, or whatever it might be. The reason that a genotype consists of 2 letters is because one letter represents the allele on one of a pair of chromosomes, the other letter represents the allele on the other chromosome of that pair. It may be big A, big P, little p. It may be big A, or big B, or little c. It could be anything. Big A represents dominant, little a represents recessive. You can use different letters of the alphabet, but you always use the capital and lowercase as appropriate. A Punnett square does not tell you all the children that parents will have. What it does is allow you to see the possibilities given to parents of the offspring that they could have. In the same way, there's a 50-50 chance of having a girl. That doesn't mean that 2 parents won't have 4 girls and no boys. So you can predict chance. What may happen could be slightly different. Instead of using big R and little r, you are using X for the X chromosome, Y for the Y chromosome. But you also need to annotate to say that there is an allele or a gene on that chromosome with different alleles that can cause potential disorders or diseases. We take haemophilia as a common example. The female has to be XX because she is she. The female is a carrier, which means that she's phenotypically normal, is not a haemophiliac. So one of her X chromosomes carries the dominant allele, so it's X big H. And you write it like this. The other allele is X little h, so that means she carries a recessive allele but doesn't have the condition. Dad is a normal male. He is XY. The Y chromosome does not have an allele for haemophilia on it because it doesn't have that gene along its length. So the dad is X big H, he's normal, and Y with no annotation. The prediction from these two parents, again, gives you 2 boys and 2 girls. One girl will be completely fine, X big H, X big H. One girl will be X big H, X little h, which we call a carrier female. She doesn't have the disease, but she carries the allele. The boys, it's a little bit more bleak. The boys are either X big HY, which means they are phenotypically normal, or X little hy, that boy is a haemophiliac and will require medical treatment for bumps, cuts and bruises. When you write the genotype ratio, you have to write 1 to 1 to 1 to 1 with the information as added. Phenotype ratio also has 4 parts to it, not just 1 or 2 or 3. If we ask you if there is a daughter, what is her chance, then it's not out of 4, it's out of 2. So be careful with the questioning. A pedigree is simply a way of representing the people that we know. Is this a dominant or a recessive condition? Shaded means disease. It's recessive because 1, 1 and 1, 2 don't have the disorder, but 2, 3 does. All right? If you know that it's recessive, then you can annotate probably most of the people on that pedigree, and that is something that will help you when you are doing exam questions. Whether or not we ask you to, you should do this as a matter of course. Here's an example I want to work through. If you're in my class, you've already seen this, so don't spoil it for everyone. You may have seen this from somebody else as well. That pedigree, is the condition dominant or recessive? It's recessive. Is it sex-linked, or is it what we call autosomal? That is, it's not on the sex chromosome. It's autosomal. Okay? We know that because it skips a generation, and because Molly's dad would have to have an X-linked condition, and he doesn't. For her to have it, she has to get X recessive from mum and X recessive from dad. Dad would have the condition, he doesn't, it's not sex-linked. If we use lettering big A, little a, we can do some working out. Mum and dad are both heterozygous. If you don't know what that word means, it is an important one. It means one of each allele. That is our Punnett square. What is the chance that Molly is heterozygous for this trait? That is big A, little a. What's the chance? 1, 2, 2 in 4 chance, 50%. Yes? No. Go back to the pedigree. What does a person who has the trait look like? They are shaded. Is Molly shaded? Does Molly have the disease? Is Molly little a, little a? No, she's not, because little a is what you would be if you had the disease and she doesn't. It means that this is actually not a possibility for Molly. It means that there is therefore a 2 out of 3 chance, and now we've got that right. We're not going to ask you to demonstrate that skill. What we are going to do is ask you questions that may require you to interrogate or extract information from a pedigree. Make sure you are taking care with that, because you may fall into traps like that as you work through the paper. I'm now going to work on evolution, and again I'm going to focus on a couple of key ideas. I wanted to summarise first to put into context some of the things that we did, and evolution was a very big ideas topic. Some of the biggest ideas were that there can be change at all. Now we take that for granted that that is possible. In Darwin's time that was certainly not an accepted view. So what sort of pressure might he have been up against with relation to this presentation of change over time? We look at how species arise, we look at how we have evidence for this, and we then turn the focus on ourselves as an example of evolution over time. You may have heard of Darwin and Wallace, you may have also heard of Lamarck. Lamarck suggested that as a giraffe reaches for tall leaves, it stretches out its neck, and that when the giraffe has kids, the long neck is passed on. If you lose a leg in an accident, will your children be born with 1 leg or 2? 2. Have you experienced, well done, have you experienced a genetic change because of that accident? No. Is there any difference to the genes that you pass on to your kids? No. Stop yelling out the wrong answer. I know that you know better than that. So we ditched Lamarck's idea in favour of Darwin's, but what Lamarck's idea did was open the door for people to even think about change over time. We are likely to ask you a question which relates to natural selection. When you answer that question, you will need to cover 4 details at least, and the details are this. Number 1, there must be variation in a heritable trait. Number 2, there must be some selection pressure making it difficult to survive. It could be food, competition for resources, competition for mates, change in environmental conditions, too hot, too cold, too wet, too dry, whatever. Some organisms must have an advantage given the features that they happen to have. It may be that it's good to be taller, it may be that it's good to be shorter, darker skinned, lighter skinned, more furry, less furry, whatever. And the last thing is that this trait must be passed on to the next generation, because the individuals that possess the trait that allows them to survive and can breed. If you die before you breed, your genes die with you. I'm now going to spend the rest of my time pulling this question apart. This question relates to a question we put in an exam 2 years ago. The crabs that exist there are the predators. We are looking at the southern and northern blue mussels. The crabs are new, the mussels were already there. The mussels are food for the crabs. The crabs crunch through the shells and eat the insides of those animals, those mussels. Over time, we see the shells becoming thicker and thicker in the southern population, but not the northern population. We relate this to natural selection now. What trait varies within the southern population? Shell thickness, be specific. What is the selection pressure? What's making it tough to survive? Predation by the crab. The crab being there is not annoying the mussel, it is eating the mussel. Be specific, please. What variant, what version of that variation gives some mussels an advantage over others? Thicker shells are a good thing to have because you get better protection from the Asian shore crab, and those that survive will successfully reproduce. Those alleles will be passed on to the next generation. What do you expect to see over time in shell thickness? Gradual increase. Would you see the same thing in the northern population? No. Is there variation in thickness up there? Is there a predator? No. Remove the selection pressure, remove the advantage, remove the change. Speciation involves 3 steps. 1, isolate the populations. Don't let them breed with each other. 2, natural selection occurs in different directions. It may be hotter in one environment than another, and different features may be favoured. 3, when they come back together, if individuals from population 1 and 2, the green and the yellow groups, can no longer interbreed and have offspring that are fertile, they are now different species. You can breed a horse and a donkey, you will get a mule. A mule cannot do anything apart from its own job, it cannot produce any offspring. Patterns of evolution I'll leave for you. Divergent and convergent. Suffice to say that for convergent evolution you will never ever get 2 species turning into one. That cannot be reversed. That's a little furry flying squirrel. It's got a little parachute in it. Fossils, biogeography, human evolution. Don't say we evolved from chimps. Chimps evolved as well. We see changes to our skulls and changes to our skeletons. The easiest one is the skulls became larger, which gave us space for brains. We became smarter, and so we survived. There are no other hominin species that currently exist. We are the only ones. Artificial selection is, instead of nature choosing what survives, it is us. If you think that it's always good for survival, think about pugs. Think about dachshunds. It's not necessarily what's best for survival, it's what we choose that survives, that allows us to change species over time. Investigations, Mr Ho.
Hi. Alright, so can you turn to the person next to you, right, and say, I am rad, see, and then give him a gangster pose. Go. I am rad, see, gangster pose. I am rad C. So when we write a scientific report, you always write it in a certain way. We always start with the introduction, then we go to the aim, method, results, and conclusion. Results and discussion and conclusion. I am rad is my kind of short form to remember all of these things. I for introduction, M for A for aim, M for method, R for results, D for discussion, C for conclusion. Now the scientific method works for everything. If you have ever wondered anything in your life, you can go, hey, the scientific method works. My words are getting jumbled. So if you've ever wondered, like, how do I get a boy to like me? I don't know. Maybe I'd go through the scientific method or these sorts of things. So I don't know. I don't know what you guys are interested in. I legit, when I was writing this up, I was legit looking up Taylor Swift and like looking up what her like most popular song was. Turned out to be the newest one. I don't remember what it was. Anyways. But that's a scientific laboratory report. You have to write something like that. Your conclusion needs to start with to determine or to do this, to do that, or to do this. So, right, your aim needs to be to do that. There are 3 types of variables that you need to worry about. The first one is your independent variables. So that's the stuff that you do to your experiment that doesn't change, that you do to your experiment. You only need to do one thing. If you're doing more than that, you're doing too much in your experiment. You can only do one. Dependent variables are the things that change as you change the experiment. So the thing that you've done to the experiment changes something in the experiment. So in this case, we've got a tree, and how high the tree grows is dependent on what you give it. And finally, the controlled variable is everything else that you don't want to change. So everything you want to stay steady will stay steady. That is your controlled variable. Now there's a few types of data. We've got qualitative and quantitative data. We've got, can you see that? Yes? Okay, and we've also got continuous and discrete data. I don't have time to go into those, but have a think about these types of data, and then you've got to think about, hey, how do I graph these types of data? What do I do with this data? So graphing data, pretty simple. This is a graph that I've looked at. This is really cool to graph. I really enjoy it. It says you have your stress level at the bottom and your performance on the right-hand side. If you have a look at it, it tells you that the more stress you get, the more you perform. But then if you get too stressed, you perform worse. You get worse. So this is just a tip for your exams. Try to keep yourself in the optimum stress level. Don't go too low, don't go too high. Okay, so in graphing data, you always put your independent variable where? On the bottom or on the side? Independent variable goes on the x-axis. So on the bottom, your dependent variable goes on the y-axis. When you describe trends, you can use a lot of words. You can use negative and positive. So if your data is going like a bell curve to one way, you call it a skew. If it's a straight line, you call it a correlation. So a positive and a negative correlation. And when you write stuff in your exams, you have to tell me about not just if it's a positive correlation or what that kind of stuff. You have to talk to me about what the graph is actually saying. So before we talked about stress and performance, so you have to talk about stress and performance in your ways. I'm just going to jump over that. So trends look like dots. You can see the trends on there, and then what you can do after that is you can plot a trend line. So that gives me a line that shows what the trend and where the trends are going. So it tells me stuff that's more than just dots. Graphic data. We all think that we can draw awesome until we start drawing. Please don't give us graphs that look like this. Please remember to use your rulers when you draw graphs. Global systems. Global systems comes in 3 main concepts. You get your cycles in nature, so that's your nitrogen cycle, water cycle, carbon cycle. You've got your greenhouse effect, and you've also got the global warming, the consequences of global warming, and we're going to go through those really quickly. So global systems, here we go, ready? Here is a diagram I drew in class with my classes. This is the carbon cycle. If you have it, bring it out. If you don't have it, you can print this one off from the slides that we're going to give you. Let's have a look at the ones that humans are actually doing to the world. So what we're doing is, first of all, we're burning excessive carbon dioxide through burning fossil fuels. And that's given. And another thing is deforestation, clearing of trees. Trees take carbon dioxide from the air and use it and turn it into sugar that we can use. So when we do that, when we remove the trees, we remove the thing that takes carbon dioxide out of the atmosphere, and that's not good. The enhanced greenhouse effect. We're going to talk about the greenhouse effect now. I'm just going to ask you a question. Which of the following factors contributes the most to the greenhouse gases? Have a think. Is it carbon dioxide, methane, CFCs, or water? Who says A? Put your hand up for A. A? A about, let's just say about half, no, 30%? Let's say who says B? B. Anyone says B for methane? Yep, a couple say B. What about C? Chlorofluorocarbons. C, no one knows what they are. That's okay. We used to use these in refrigerators. And water. Who thinks water? The correct answer is actually water. Water actually contributes to the greenhouse effect the most. But that's not a man-made effect, so we're just going to talk about the greenhouse effect with what we do. The greenhouse effect happens naturally, so as you can see, this is our Earth, it's got an atmosphere around it. There's energy coming in from the sun. The energy that comes in from the sun strikes the Earth's surface, and it changes the energy from infrared or light energy into heat energy. And this heat energy sometimes it gets reflected back out into the atmosphere, and the atmosphere will usually absorb it and keep the Earth warm, so we don't freeze to death, and it will help us live. But what we've been doing is we've been pumping out a lot of greenhouse gases into the atmosphere, which causes an enhanced greenhouse effect. So that makes it even worse. So our atmosphere is here to keep us warm, but if we keep applying and keep giving it more and more greenhouse gases, it makes the Earth warmer. And there are lots of consequences for that. So when we decrease carbon dioxide, the deforestation makes it, did you see the animation there? The arrow got smaller. Carbon dioxide decreases. I'm just going to keep jumping. Carbon dioxide, we keep pumping carbon dioxide into the atmosphere, and there is no way for us to get rid of it at the moment. Another impact is that because sunlight gets reflected off white surfaces, polar ice caps reflect a lot of light. They don't absorb energy, so the planet doesn't warm up as fast. And so when ice caps melt, we show a lot more ground for the sunlight to hit the Earth's surface and warm it up. Another thing is rising of sea levels. As you can see here, 1 meter in rising sea levels will cause Venice to be underwater, 2 meters Amsterdam, 3 meters about Hamburg and St. Petersburg, about 3 meters Los Angeles, San Francisco and Lower Manhattan will all be underwater. So we have to really consider our greenhouse emissions. Another thing is from permafrost. Permafrost is frozen ground that's been frozen since the last ice age, and there's a lot of carbon in there. So if we freeze that, carbon comes out, causes more greenhouse effect increases, more temperature, more melt. Bad. Impact of climate change. Another thing is extreme weather. Extreme weather our weather is caused by our atmosphere. If we play with our atmosphere, we cause changes in weather. And extreme weather patterns have been seen all around the world until today. And most recently as well. And last of all is the impact of climate change on life. Biodiversity. If we change the climate, certain animals live in certain tolerance ranges. If I stuck you in a volcano, you would burn to death. If I put you in space, you would have no air to breathe. That's not an environment you can live in. So when we change all the environments for the animals, they will die out as well. And we will lose our biodiversity because of climate change. And that's all I have for you today. Mr. Hick.
All right.